Good morning, all. Thanks for joining on this Monday morning. I hope you're doing well. First, I want to thank you for participating on Friday. That discussion turned out to be a gold mine for good jokes and memes, and I had a good laugh. I hope you had time to go through it and have a good Friday as well. Have a pleasant Friday as well. And if you haven't, go back and see some additional jokes posted because people kept posting up until midnight. All right, so are there any questions before I start the lecture?
Did you watch the Super Bowl?
Ah, no, I'm not a football fan. I'm a soccer fan. But I know who is it? Tom Brady is a huge deal. I hope I got the name right. And he was on top of his game. I watched the news this morning, and that's what they told me. So that's all I know about Super Bowl. Oh, not everybody likes Tom Brady. Okay, I know Giselle Bundchen, who is his wife. She's a model from Brazil, I think. Anyway, any other questions? Horse related? Okay, let's get started then. So, sharing my screen. Okay, I got a question related to the study guides, and Ansley is finished with the study guide. She should email you today, probably. Ansley, are you on? Can you update us on the study guide?
I am. So I finished it. It's good to go. I will post it after class in announcements. And I'll go ahead and send an email for anyone that's not here. But you should be able to find it under announcements after class.
Great, thank you, Ainsley. Okay, let's see what other questions, if there are any. Okay. So I created two participation assignments for today. One is for 8 points and the other one is for 2 points. Although like this assignment is the participation assignments for testing the lockdown browser is it's not real questions, okay? I just want to test the lockdown browser. You will get 2 points for participation in it, but it's just literally select true from below or select false from below. But it will allow you to get familiar with the lockdown browser and anticipate any issues or any additional time that you will need for the exam. So this is a low weight participation assignment that is designed to test for you to get familiar with the lockdown browser and so that you don't have problems during the exam. And so this participation exercise, the testing one, is you can take it between today and Wednesday, complete it between today and Wednesday. And the participation assignment for 8 points for mitosis and sexual reproduction, it's going to be due today. All right, so that's the participation, is where we stand on the participation assignments. Quiz 3 is open and is due on Friday, February 12th. Okay, Jana, did she say 2? I only see 1 for today, or did I miss here? There are 1 is due today. I posted another participation assignment designed to test the Respondus lockdown browser. And it's got 2 points, and you can take it between today and Wednesday. So you have 3 days to test Respondus lockdown browser. Please complete it, because otherwise you might have issues during the exam. Okay, so quiz 3 is open. You can take it anytime between today or last weekend, and some of you did, good job, and Friday, February 12th. All of the material covered in quiz 3 we already went over. Okay, so it shouldn't be much new stuff apart from the supplementary videos which I posted in the quiz description. And quiz 4 opens on February 13th, and it is due on Monday, February 22nd. So we're going to change the pattern when the quizzes were due. So before they were, the past 3 quizzes were due on Friday, but this quiz 4 is going to be due on Monday. But it's not until like next week, so don't worry too much about it yet. And as I mentioned, there are 2 participation assignments. One is due today and the other one is due on Wednesday. And the testing lockdown browser is kind of a dummy participation assignments, but for real points. So you will not get it wrong. You will not lose those points. All right, so last time I covered the DNA replication and binary fission. So DNA replication happens before a cell divides. DNA replication is needed by the cell so that when the cell splits, the daughter cell will get equal amount of DNA in each other. So binary fission is the simplest type of cell division, and it's characteristic for prokaryotes, for individual single cell organisms that do not have a nucleus. Now, the eukaryotic cells, or cells with the true nucleus, they divide via the process called mitosis. And I'm sure you've heard the term before and even studied it or attempted to study it. Well, if you did, we're gonna go through it again. So mitotic cell divisions allow an organisms to grow and develop repair tissues and regenerate body parts. So mitosis has many roles. It allows organisms to grow and develop, repair tissues. For instance, if we scrape our knee, the wound will heal because our skin cells divide mitotically, and maybe some scar tissue will remain. But the point is, the body repairs itself. If we sometimes people damage their liver with medication or alcohol. So if the damage is not too bad, actually liver is one of the few organs in our body that can regenerate itself. So as long as you follow doctor's advice, there is a good chance that the liver can regenerate. But not all human cells divide at the same rates. So skin cells divide very rapidly to compensate for damage. And I mentioned during last lecture, I believe that the turnover time for skin cells is about 35 days. So every 35 days, you end up with a whole new skin layer. So none of the skin cells that you have after 35 days are the same. But some cells within our body do not divide as rapidly. For instance, the nerve cells, mature brain cells, do not divide rapidly at all. And they just kind of stay at the point that they are. So if you get brain damage, the cells will not regenerate. They will not divide mitotically. So what brain will do often is just rewire its wiring, pardon the tautology, to compensate for the lost functions, to compensate for the lost areas responsible for some functions. Person's age affects cell division rates. The younger the person is, the faster the division rate is. And as people get older, the processes within their body, including mitosis, become less efficient, and things go slower. So mitosis, though, it's just one part of the cell cycle. There are 3 additional parts of the cell cycle, which I will discuss next. So the cell cycle is a repeated series of events, and there are technically 4 stages of the 2 big stages of the cell cycle, and 4 kind of sub-stages of the cell cycle. So the 2 big stages of the cell cycle are called interphase and cell division, or mitosis plus cytokinesis. During the interphase, the cell just goes about its business, does its job, whatever its function is. If it's the what is it called? Pancreatic cell, it will generate insulin. If it's skin cell, it will express melanin if you're in the sun, and so on. During the mitosis or cell division, as you can guess, the cell divides. So the cell either exists or it divides. That's the metaphases. Now the interphase is divided into 3 sub phases. So G stands for gap and S stands for synthesis. during the G1 phase, the cell has just divided, so it's a newly divided daughter cell, and it performs its normal cell functions and grows. Okay, so if it's pancreatic cell, it will produce insulin. If it's some other cell in the body, if it's a cell in, I don't know, intestinal cell, it will produce some enzymes and whatnot. Well, no, enzymes are produced by pancreas. Never mind. During the S phase, though, when the cell enters the S phase, which again stands for synthesis phase, and the synthesis indicates the DNA replication. So the cell at this stage, the cell replicates its DNA. So it was personally for me, it was kind of hard to remember that the DNA replication is separate from mitosis process. So first the DNA replicates and then the cell divides. So DNA replication happens actually during the interphase. It does not happen during mitosis. So the synthesis of DNA, again, happens during interphase, but during mitosis, this DNA is pulled apart into the two cells, okay? After DNA is replicated, the cell enters G2 phase or GAP2 phase. And it has some additional growth, and it synthesizes the stuff necessary to enter mitosis stage, or it synthesizes some stuff necessary to prepare for division. Now, mitosis is after the GAP2 phase, the cell enters the cell division metaphase, and which is mitosis and cytokinesis. Mitosis consists of 4 stages: prophase, metaphase, anaphase, and telophase. And I believe the next slide will talk about that. Yeah. And after the 4 stages of mitosis, the cell enters the cytokinesis stage, which, if you know Greek, cyto means cell and kinesis means movement. So during cytokinesis, the cells actually move apart. One cell becomes one parent cell becomes 2 daughter cells. Okay, next I'm going to discuss the stages of the mitosis in more detail. But before I go there, are there any questions? Okay, moving on. So mitosis is a series of steps. Mitosis is divided into 4 phases, as I just mentioned before, and plus cytokinesis. So the 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. And cytokinesis is literally splitting of one parent cell into 2 daughter cells. So mitosis starts with the prophase. During the prophase, DNA coils very tightly, making chromosomes short and thick. Remember, during the previous lecture, I discussed the DNA division, DNA synthesis, and I mentioned that the chromosomes are visible in the microscope in their classical X shape only when the cell prepares for division. So during the prophase, the cell still contains, during early prophase, the cell still contains the nucleus. And if you look in the nucleus, you will see the classical X-shaped chromosomes, which we often associate with the DNA shape. But before the cell enters the prophase, the chromosomes are just loosely packed within the nucleus. They're not in X shape. It's very hard to distinguish the shape of a chromosome before the prophase. So during the prophase, at the same time, a structure called the spindle forms, and it is made of microtubules. So here are this purple whiskery blobs here are called centrosomes, and it's one of the organelles in the cell which I didn't touch upon before. It's going to be maybe new to you, but the function of these centrosomes is to form the protein fibers which will pull apart chromosomes later on when the nucleus dissolves. So this is a more detailed picture of what happens during prophase. So here's our late interphase, where when the cell prepares to divide, prepares for mitosis. And you can see that if you look in the nucleus during late interphase, you can see that the DNA, the chromosomes are not X-shaped. So chromosomes are not DNA is not tightly packed into chromosomes in an X shape. Now, during the early prophase, we see that the centrosomes start growing these spindle fibers, which will help during the late prophase. The nucleus during early prophase is still intact. We can still see the whole nucleus during early prophase, but as we, in the late prophase, as we move along in the cell cycle, during the late prophase, the nucleus dissolves, the nuclear membrane dissolves, and the spindle fibers grab onto chromosomes and they kind of hold on to them at this point. And this is when late prophase is considered to end. So what are the characteristics of the prophase stage is the spindle fibers form, these protein structures called spindle fibers form. The chromosomes become X-shaped, meaning the DNA is more tightly packed. The nucleus dissolves and the spindle fibers grab onto chromosomes. So all of these are characteristics of the early prophase. So once the spindle fibers grab onto chromosomes, the cell enters the metaphase. And during metaphase, these purple spindle fibers, they align the chromosomes along the equator or the middle of the cell. And the two chromatids, and chromatids, remember, are the half of the chromosome. The two chromatids are still held together by the centromeres or by spindle fibers, okay? And this is the metaphase. So metaphase, the characteristic of metaphase, if you were to look in the microscope and ask to identify which stage of mitosis are you looking at, if you saw the chromosomes lined up along the center of the cell, you would say, and the chromosomes were still like X-shaped, you would say the cell undergoes the metaphase stage of mitosis. Now, the third stage is anaphase, and during the anaphase stage, the two chromatids, and remember, chromatids are one half of the chromosome. Chromatids split and are pulled apart into each half of the cell. So was the spindle fibers, they shorten, and because the spindle fibers shorten and pull back to the centromere or centrosome, the chromosomes are pulled apart to each half of the cell. And the final stage of the mitosis is called the telophase. And essentially, it reverses the events of the prophase. During telophase, the spindle fibers shorten and centrosomes become small again and move to the polar opposite side of the cell. The nuclear membrane forms and it encloses the chromatids, the DNA essentially that was pulled apart into different parts of the cell. And so now we have a cell with the two nuclei, and the last part that is left, what is left is basically for the cell to separate, for one cell to be split into two, because one cell should not have two nuclei. And this phase or the stage where the cell is pulled apart, or one cell splits in two, is called the cytokinesis. So the cytoplasm and the two nuclei are distributed into the two daughter cells, which are physically separate, and they're separated by when this contractile ring basically forms in the middle of the cell, and the contractile ring, literally as the name suggests, contracts, and the two cells separate. So contractile ring, basically it's a cell wall that is built in between the two cells. In the animal cells, things go a little bit differently. In the animal cells, the so-called cleavage for furrow is formed between the two cells, and the contractile ring forms from protein, actin, and myosin, the same two proteins that are in our muscles. And this contractile ring basically contracts and separates the two cells. So what is pictured here is the contractile ring, meaning these are two animal cells. And if in the division of the two plant cells, the cell wall is just built in between the two cells, okay? And after cytokinesis, the two daughter cells enter GAP1 stage, or early interphase. Okay, any questions about that? I'm sure all of the information I've gone through is already familiar to you, but if you still have some questions, remaining questions, now basically is the time to ask.
Yeah, I have a quick question.
Go
So
ahead, Madeline.
you said that plant cells have the contractile ring and the animal cells separate differently?
Sorry, it was vice versa. The plant cells build the cell wall in between the two daughter cells, and that's basically how they separate. The plant cells, the plant grows, right? The plant cells don't separate very far from one another. So it's just one cell when one cell has two nuclei, when it's undergone division, the plant cell wall is built in the middle of the cell with the two nuclei. But in animal cells, they separate using the contractile ring. And contractile ring consists of the two proteins, actin and myosin, the same as in our muscles, which contract and separate the two cells.
Okay, thank you very much. That was very helpful.
All right. Good. Any other questions? All right, moving on. The cell cycle is under tight control. There are a series of chemical checkpoints that regulate the cell cycle. So several internal checkpoints ensure that a cell doesn't enter one stage in its life cycle until a previous stage is complete. So these checkpoints, they screen for damaged DNA, and some signaling protein may trigger apoptosis or cell death. So if DNA is not replicated correctly, if there are some mutations introduced during DNA replication, the cell is killed. And I believe somebody posted a meme about that. That was pretty funny. All DNA is replicated, and I don't know where was it going with that? I don't remember anymore. Oh, okay. So checkpoints ensure that all DNA has replicated and was replicated correctly. And if it was not, the cell is killed or undergoes apoptosis. The checkpoints also ensure that DNA is not damaged. And if it is damaged, the cell is killed. And the checkpoints also ensure that chromosomes line up and separate properly. And if they don't, the cell is what? The cell is killed. The cell is it undergoes the programmed cell death, so it undergoes apoptosis. Okay, I have an example here in my notes, which I forgot about. I'm not going to go into it because I forgot about it. I don't remember what I was going to say. So, an example of mitosis gone haywire is basically cancer. So cancer cells divide out of control. So technically, the cancer cells are doing what the cells do. They undergo interphase and undergo mitosis, but the mitosis is uncontrolled in the cancer cells. So the body loses control of cell division, and that's basically what it is. Like for me, it was pretty neat to find that out. I mean, it makes it less scary if you understand what's going on. So cells continue to divide when they do not need to overwrite the checkpoints. So when the checkpoints fail in the body, that can lead to cancer. There are some tumors that are not cancerous, and they are also considered to be dividing kind of out of control. So an example of a tumor that is benign is a mole on the skin. Okay, so some benign mole, and in a mole, technically, some cell, when it replicated, it had either a mistake during the DNA replication, or some external factor damaged its DNA. So maybe UV rays, the sun rays damaged the DNA of a skin cell, and the skin cell started to divide out of control, creating a mole. So the mole can be benign or malignant. And in the benign mole, basically there is a capsule around the mole that prevents the cells that are dividing kind of out of control from escaping the particular location. So the cells within the mole stay in the mole. Whereas if the mole is malignant, these cells that are dividing out of control, they will not be contained to the site of the mole. They will get into the lymph system and then into the blood and circulate all around the body, kind of seeding these malignant cells in various parts of the body, which is called metastasis, technically cancer metastasis. So an example of the benign growths that are encapsulated by this capsule and which prevents the cells from escaping a particular location of the cells that are dividing out of control are warts and moles. And yeah, basically the malignant cells do not have this capsule, and this is what is dangerous about the cancerous growth. They do not have a mechanisms that would contain the cells that grow out of control to the particular site. The cells spread around the body, and the growth starts in new places. There are plenty of treatments for cancerous growth. So cancer treatments include surgical tumor removal, and sometimes it's enough if the cancer stage is in its early stages or in the stage before the cells spread to the new locations. Some anti-cancer drugs can slow or block cell division. Unfortunately, these drugs also affect other cells that divide rapidly. And an example of cells that divide rapidly is the cells that produce hair. So that's why when people undergo chemotherapy, they kind of lose hair because along with this fast-growing, fast-dividing cancer cells, the drug targets other fast-growing, fast-dividing cells, or like hair cells. And also the third type of treatments that is widespread is radiation treatment. So it may seem counterintuitive that, because I just discussed a couple of lectures ago that radiation actually mutates DNA and it can be harmful, because if the DNA is mutated, the mutation can be such that leads to uncontrolled cell division. But if you radiate specific type of cells or a specific location on the body, like where the cancer is, then it has a high probability of just killing only the cancer cells. So radiation treatments are often highly localized. Not the whole body is irradiated, it's just the specific area of the body where the cancer is located, and the radiation beam is very narrow, is very specific. So that is it for mitosis. Are there any questions before I move on to sexual reproduction and meiosis?
So there's a really interesting question about benign tumors and if they always stay benign or if they can become malignant.
Oh, good question.
Not sure.
Sometimes the skin growths, in particular, they're widespread. So sometimes the skin growths can become malignant. Doctors recommend checking these discolorations on the skin and keeping an eye on them. So like dermatologists, especially here in Florida, right where it's the Sunshine State, dermatologist recommends like watching your moles and see, and if the mole starts changing in shape, in color, or if it starts bleeding, go to the doctor, because those are the signs that the mole is turning from the benign to the malignant one. I hope I answered that question or gave one example. So Blaine asks whether I could go over the G phases. Sure thing. Let's go back here. So interphase is divided into 3 phases. G1 phase, GEP1 phase is the phase when cells function normally. So the cell grows and some protein synthesis occurs. Like if it's a cell in the pancreas, it will produce insulin and other enzymes that help with our digestion. If it's a good God, hepatic liver cell, if it's a liver cell, it will produce some enzymes that will help us digest fats and so on. So in the GEP1, during the GEP1 phase, the cell grows and performs its normal functions. During the S phase, which stands for synthesis phase, DNA is replicated. So DNA is basically duplicated as we've discussed in the previous lecture, in the way that we discussed in the previous lecture. And during the GAP2 phase, the cell has some additional growth, but mainly it prepares for the prophase. It grows the spindles. It synthesizes some proteins that are necessary for cell division. And this is basically the summary of the interphase. So during interphase, when the cell is not dividing, that is the interphase. That is literally the definition of the interphase. When the cell is dividing, it undergoes mitosis and cytokinesis. So GEP1 phase, normal cells growth and cell growth and function. S phase, synthesis phase is DNA replication. And GEP2 phase is preparation, synthesis of some stuff that is necessary for cell division. So spindle fibers, Alexander asks question, do spindle fibers form in G2 or prophase? The answer is both. So during late G2 phase, they start to form, but during prophase, they grow. They become really long. Okay. Any other questions before I move to sexual reproduction and meiosis?
I was gonna ask, what was the really is there anything super important that we have to learn from the cancer section? Because you briefed over all of it and I kind of understood those concepts, but is there anything we really have to focus on?
Well, the main concept that I would like you to take away from this lecture is that cancer is uncontrolled cell division. It's I don't know, personally to me it helps kind of understand it rather than demystify what it is, and understand that not all cell division is cancerous. So what is the difference? Like what makes a growth malignant? And the answer to that is the absence of a capsule that would contain the cells that are dividing uncontrollably. Okay, so if there is a capsule, then the growth is benign. If there's no capsule that contains the cells that divide uncontrollably, then the cancer is malignant. The growth is malignant. Did I address your question, Dylan? You kind of cut off there. Okay. Any other questions? Good. All right. So our next topic is the sexual reproduction and meiosis. And meiosis is another type of cell division, which is very specific to sexual reproduction. Meiosis happens only during sexual reproduction. So our skin cells divide mitotically, but the cells within our, well, if you're a woman, within your ovaries, they divide meiotically. So this sexual reproduction happens in both plants and animals. And in this picture here, you see a bee assisting a plant to sexually reproduce. So the bee gathers the pollen, which contains which contains gametes, which are basically gametes, and it distributes these gametes to other flowers, and thus pollinating them, or introducing male gametes to female gametes in another flower. So it aids the sexual reproduction of the plants. Asexual reproduction, A meaning not. Okay, asexual reproduction is relatively rare. Most of the organisms that we know of, they reproduce sexually. And the examples of the organisms that reproduce asexually are basically single cell organisms. And if an organism reproduces asexually, it just basically undergoes mitosis. It does not undergo meiosis, it undergoes mitosis. So all of the genetic information contained in the parent cell will be inherited by the daughter cells. So in this picture here, you see the amoeba that undergoes reproduction cycle or undergoes mitosis, and that's how it reproduces. So the two daughter cells will contain the exact same genetic information as the parent cell of amoeba, as a parent amoeba. So if the reproduction is asexual, the offspring will have identical genotype to the parent. But the sexual reproduction, like these, the dogs here, the pups were produced by sexual reproduction. And you can see that the pups, they do not have the same color as the mom. So that indicates that the offspring, if in the sexual reproduction, the offspring do not have the exact same genotype as the parent. So some of the organisms that you're well familiar with, like plants, they undergo both type of reproduction. They can reproduce sexually and they can reproduce asexually. So if you want to grow a plant from a parent plant, you can just take a leaf out of a plant, stick it in water, and you will see that the roots will form eventually, if the leaf will not die. And when the roots form, you can just plant it in some soil, and the new plant will grow from that. So this is an example of asexual reproduction. Okay, nothing sexual was going on. You just took one piece of plant, stuck it in water, and then planted it in soil, and it will grow. And plants can reproduce like that, or they can reproduce via just root growth, below ground growth. So roots can spread and new plants can emerge from the spreading root system. And that is an example of asexual reproduction. And plants can reproduce sexually, as I showed in an image previously, when the bees or pollinators help spread plant gametes among the individuals. So for sexual reproduction, a special type of cell division is required, and the special type of division is called meiosis. It is a type of cell division that generates sperm and egg cells, or gametes, each of which is genetically unique. So during meiosis, some serious mixing up of the genetic material is going on, specifically to generate this genetic variability in the gametes. This genetic variability is highly beneficial. This genetic variability in gametes produces genetic diversity. And from the evolution standpoint, this genetic diversity is very, very well worth it. Although the sexual reproduction can take a lot of energy and is risky, because of the resulting genetic diversity, it is worth it from the evolutionary standpoint. So what is the energy needed for? So attracting a mate and producing sex cells requires a lot of energy, which is costly to the organisms. Moreover, attracting a mate may not be successful. So although one individual may not be successful in reproducing sexually, on the population level or on a species level, the genetic diversity produced by the sexual reproduction is still worth it, even though one specific person may not have benefited from it. So I want to stop at this genetic diversity thing and give you an example of how illustration rather, not an example how it's worth it, but illustration that the sexual reproduction is advantageous. So one example that can be illustrated from one example that illustrates this focuses on reproduction of a roundworm. So a roundworm shown here, it can reproduce in two ways, sexually and asexually. So scientists took advantage of this roundworm and its two ways of reproduction and conducted an experiment. They exposed this worm to live and dead infectious bacteria and observed how worms reproduced over generations in dishes with live and dead bacteria. So they took a bunch of worms, they stuck them in a bunch of dishes. Some dishes contained live bacteria, some dishes contained dead bacteria. And the scientists observed in which way the worms reproduced. Did they reproduce sexually or asexually? So what are the results? So over time, over many generations of these roundworms in dishes with live infectious bacteria, more worms were reproduced sexually. In dishes with a dead bacteria, more worms reproduced asexually. So this is pretty fascinating. So in graph here, it shows basically generation 0 of the worms, generation 4 of the worms, and generation 30 of the worms. And the y-axis is the percentage of sexually reproducing worms. So in generation 0, very few worms reproduce sexually because sexual reproduction takes energy and it is costly. Okay, so it's much easier to reproduce asexually for the worm because it does not take a lot of energy. But in the dishes with the live bacteria, more worms started reproducing sexually compared to the dead bacteria, compared to the worms with the dead bacteria. And in the generation 30, this difference between sexual and asexual reproduction became even more vast. So when there is a threat to the worm, when there was a threat to the worm, to the worm's survival, they preferred to reproduce sexually compared to asexually. So why so? To create the genetic diversity, to increase the probability of such genotype that would be resistant to the bacteria. Okay, so whenever there is an external threat to the survival of the species, if the species can reproduce sexually or asexually, the species will prefer to reproduce sexually, basically, or die. I mean, one of the two. All right, are there questions about this example here? Why sexual reproduction is advantageous? Okay. So back to, let's go back to the chromosomes here. And during meiosis, the chromosomes kind of pair up and they align during one of the phases of meiosis. But before discussing all of the meiotic stages, I wanted to touch on some terminology here. And one of the terms that I want you to know is what is the homologous chromosomes. So the homologous pair of chromosomes, they have the same size and structure and contain the same genes. But each member of the pair comes from a different parent. So you see here, you see depicted two homologous chromosomes, meaning they have the same genes, maybe not the same versions of the genes or alleles, but the same genes that are responsible for the same kind of proteins. And the pink one came from the mother, and the blue one came from the father. So in our body, we have 23 pairs of chromosomes. And these, in each pair is homologous, meaning we inherited one chromosome from the mother and the second chromosome from the father. So in each of our cells contains 23 pairs of chromosomes, when one member of the pair was inherited from the mother and the second one from the father. So diploid cells, meaning cells with full pairs, contain two sets of chromosomes. Sexually reproducing organisms consist mainly of diploid cells, or 2N cells, which contain two full sets of chromosomes. One full set of chromosomes is inherited from each parent, as I mentioned before. So one pair member is inherited from the mother, and the second pair member is inherited from the father. So we have 23 pairs of chromosomes, but technically not all of them are homologous. Like if the 23rd chromosomal pair is X and Y, if you're in human males, the 23rd pair of chromosomes is X and Y chromosome, whereas in females, it's XX chromosome. So in males, the 23rd pair of chromosomes is not technically homologous, because they're not the same size, and technically they do not contain the same genes. But during meiosis, they align together, and that is why they're still considered, they're often called homologous. All right.
Are the
And
females 23rd chromosomes homologous then?
yes,
They're both X's? Okay.
yes. As I mentioned a little bit earlier, homologous chromosomes carry the same set of genes, but the specific kind of these genes or alleles may differ. So, what do I mean by that? Basically, the slide explains that the nucleotide sequence is not the same within the gene. So a gene, let's say it can produce, it can encode the eye color, but not everybody has the same eye color. We can have blue eyes, some people have brown eyes, some people have green eyes, and some people have, well, no colored eyes, like albinos. So the gene that encodes the eye color is the same, but the allele, the sequence of the nucleotides, may be different, and the gene with a different sequence of the nucleotides are called alleles. So each chromosome in a homologous pair has genes for the same traits, and the genes are arranged in the same order. So let's say we have a homologous pair of chromosomes here. We may have one inherited from the mother and the other one inherited from the father. The alleles are denoted as capital A, uppercase B, lowercase b, and lowercase d. So the alleles can be identical. For instance, the mother and the father can have both brown eyes, so the offspring will have brown eyes as well, because it's a dominant trait, dominant allele. Or let's say a mother can have brown eyes and the father can have blue eyes, so the alleles will be different. And I guess you will know from school that the child will likely have the brown eyes because the allele for the brown eyes is dominant.
Professor, before we move on, there's a question about explaining homologous versus diploid one more time.
Okay, so homologous chromosomes are the chromosomes that have the same type of genes but maybe different alleles. The homologous chromosomes are chromosomes that align together during the meiotic cell division. Now, diploid cells, they have two members of the pairs. Diploid. How do I explain this better before going into meiosis? Let me think for a bit. When a cell has a chromosome from mother and chromosome from father, it is diploid. When it has two chromosomes within a pair, it's diploid. When a cell has only one chromosome out of the pair, it's haploid. So di means two. Think if there's two chromosomes with the same genes, the cell is diploid. If there is only one chromosome with a set of unique genes, the cell is haploid. I think it will become more clear once we go over the stages of meiosis, because I will show explicitly during meiosis when the cell is diploid and when the cell is haploid. Okay, and it's 10:20. So any last minute questions before we end? All right. Well, thanks for joining, and I'll pick it up with meiosis and patterns of inheritance on Wednesday. See you guys then. And don't forget about the participation exercise.